I would like to thank this invitation for Dr. William Weisman, and I would like to invite Dr. Nancy Monteiro-Riviere. She belongs to the University of North Carolina. She's Center of Research of Toxicology of the United States. I would like to remind the speakers that we have 25 minutes for the presentations, not to compromise the seminar itself.
Well, thank you for the invitation for the organizers to ask me to present some of the research we've been conducting in our lab. As I said, I'm at the Center for Chemical Tox Research and Pharmacokinetics at NC State University. We heard some previous speakers today speak about inhalational toxicology and the effect of nanomaterials, but there's other routes of exposure that we should think about, because when nanomaterials are generated from a source, they go into the air, but then they also come down and then also can come into contact with the clothing, hands, and shoes, and also during occupational and manufacturing. There's an opportunity to come in contact with nanomaterials. This is a research lab in which the students producing these nanomaterials did not even wear gloves or a respiratory device. So when you look at whether an occupational scenario or in cosmetics or in sunscreens, nanoparticles are becoming ubiquitous in a lot of our products today. And if you look at where the protective PPE that they're wearing, you can see there is an interface of skin that's exposed between the glove and the coat, so you can see that skin becomes an important route of exposure. So we know that the biological impact and the biokinetics of nanomaterials is dependent upon these physicochemical properties: size distribution, agglomeration state, shape, crystal structure, chemical composition, surface area, surface chemistry, surface charge, and porosity. And all of those physicochemical parameters are responsive on how these cells get taken up, how it affects protein binding, and also how it can interact with your cells, your body fluids, and proteins that would have the ability to distribute throughout the body. So today I'm going to talk a little bit about skin as an important organ and the effects of nanomaterials on the skin in both in vitro cell culture studies, in ex vivo studies, and in vivo studies, where we've looked at many different types of nanomaterials. But skin is a very complex and dynamic organ consisting of an epidermis and a dermis, and a very upper layer, stratum corneum of the epidermis, which is a non-viable layer that represents the major rate-limiting barrier to chemicals and nanomaterials. If this barrier is breached somehow, then particles can come through, trigger a cytokine response or an immune type response. But particles or chemicals can come through the cells, between the cells, through openings of hair follicles or opening of sweat ducts. So if you look at this upper stratum corneum here, it sort of looks like a brick and mortar, where the bricks represent the corneocytes and the mortar in between, in which particles or chemicals travel, is your cement or your mortar. So once this area is breached, then particles can penetrate and irritate the cells and cause toxicity. We also know that these keratinocytes in the epidermis can function as environmental signal transducers that can convert exogenous stimuli into the production of cytokines. In our lab, we study many types of cytokine response to nanomaterials. But one thing I want to point out is among these keratinocytes and non-keratinocytes, like Langerhans cells, which are your immune target cells that can ingest particles, bring them down, and then take them to the lymph node. So if the stratum corneum is breached, then there is a reason to wonder about the safety of nanomaterials in skin. Usually cytokines can regulate different type of ICAMs, ELAN, and VCAMs that would trigger your erythema and edema. So today I'm going to talk to you about some of the work we did with multi-walled carbon nanotubes. And we knew that within 24 hours, almost 59% of the cells uptake these non-functionalized carbon nanomaterials. And by 48 hours, almost 84% of the cells had uptaken these carbon nanotubes. And these multi-walled carbon nanotubes look very similar prior to the experiment and then after the culture experiment. We also evaluated some C60s. These C60s were functionalized with a nuclear localization signal and a FITC labeled, and we realized when they were in the keratinocytes, they came very close to the nucleus. We've also shown that these keratinocytes can engulf these nanomaterials. This is a fullerene-based amino acid working with the group at Rice University, and you can see that the phagocytosis of these nanomaterials are taken into cells. So now we know that nanomaterials can easily be taken into cells. We want to know the mechanism. How do nanomaterials enter cells to trigger these type of cytokine responses and toxicological responses that other authors have presented? We evaluated with 24 different endocytic inhibitors for micropinocytosis. We evaluated clathrin. We also looked at the PARs. We looked at caveolae lipid rafts, and also looked at gene-protein couple-related protein and lipodensity receptor, scavenger receptor proteins. We studied these in quantum dots because they naturally fluoresce. And what we found and published in Toxicological Sciences is that the primary pathways in which these quantum dots enter into the cells is through the lipodensity lipoprotein receptor, scavenger receptor pathway, and also through the gene-protein couple receptor pathway. So when you work with nanomaterials in tissues, how do you find nanomaterials in tissues? It's not very easy. It's not like in cell culture, but you have to work with them either if they're radiolabeled or somehow modified or functionalized to a FITC label or some type of fluorescent tag. Otherwise, it's like looking for a needle in a haystack. So today I'll talk a little bit about absorption and penetration, and absorption relates to the amount of chemical or particle that would penetrate the skin and then get through all those epidermal layers to get taken into the blood vessels of the dermis to have a systemic effect. When you talk about penetration, penetration remains how much of the particles or chemicals get into the skin that could be available for local cutaneous activity, and we can detect that through confocal microscopy, transmission electron microscopy, special stains, and biopsies and tape strips. So we studied dermal absorption. This is classical safety assessment, or assessing penetration or absorption of chemicals is through two different methods, which is the flow-through diffusion cell or the static diffusion cell, which we've used in our lab for over 20 years. What you do is you dermatome the skin, you place the skin in here, you then dose the surface, and you look for the study of chemicals coming through and estimate the time or if a chemical does come through. Usually this is done by radiolabeled compounds. And this is a setup in the lab where you see that you have many different wells and cells where you dermatome the skin, you topically dose with your nanomaterials, and you measure for what comes through. So we worked with Dr. Vicki Colvin, where she provided us with some Q-dot 621, which are very different shape. Unlike spherical and elliptical type of Q-dots, these are nail-shaped with a peg-coiled coating. These were studied in those diffusion cells and dermatome skin at different concentrations, and you could see that we saw the Q-dots primarily residing in this upper layer of skin at both of these concentrations. This is the DIC mode, fluorescence, and fluorescent overlay. But I want you to pay close attention to the hair follicle. Well, it appears that it's in the follicle, but this can be misleading because as an epidermis, the hair follicle is an invagination of the epidermis, and I'll go into that detail a bit later. We also saw penetration of these Q dots, where you could see it in this intracellular lipid layer, and if you go in higher mag, you can see the identity of the shape of these Q dots, which are nail-shaped. So they did penetrate a little bit, but tremendously remained up in the stratum corneum layers. We also wanted to know, in an occupational setting, if you do repetitive motion and hand motion and constant rubbing and grinding motion, if flexion would cause an increase in penetration of nanoparticles. We dermatome the skin, place it on an apparatus with a 45-degree angle, flexing 20 flexes per minute, and then take these dosed skin with the Q dots and evaluated it in flow-through diffusion cells for 8 and 24 hours. The graduate student flexed it every 15 minutes through 90 minutes, and what we first saw at 60 minutes was that this FITC label basic amino acid fullerene, which is approximately about 3.5 nanometers in size, was able to penetrate with 60 minutes of flexion over 8 hours. And with 90 minutes of flexion, you could see penetration of these small particles coming through the skin and getting as far into the dermis. And of course, over 24 hours, you had more of an exaggerated type of response. We also worked with some silver nanoparticles of 3 different sizes and shapes. We have a 20, 50, and 80 unwashed. We worked with 20, 50, 80 washed particles, and also carbon-coated particles. This paper was published in Environmental Health Perspectives, in which we showed that in the unwashed particles, which are the ones that are commercially available, tended to be toxic versus the washed particles. We also do our own dynamic light scattering measurements because sometimes they can vary from the manufacturer's suggested management. We also do TEM, particle concentration, and zeta potential. What we've shown here with all the different particles we looked at, they were very homogeneous, and you can see their size. We also noticed uptake in our HEK system, the human epidermal keratinocytes, of all the different sizes. It didn't matter if they were washed or unwashed or carbon-coated or size, they all were able to be taken up into these cells. We also did an in vivo study with those same type of particles where we dosed every day for 14 days on pig skin to look for particle penetration. And to our surprise, with both washed and unwashed, they all tended to remain on the surface of the skin, that these particles did not penetrate even after 14 days through, but we did get some type of inflammatory type responses by H&E. We also verified that these particles that were seen in our cell culture and in our in vivo on the surface, that they did show peaks of silver, so they were our silver particles. Now, when we do nanomaterial absorption and penetration studies, it's important to use the right animal model. We'd love to use skin every day, but it's difficult to obtain human skin. Our human skin is obtained from surgical abdominoplasties. Within 30 minutes, we can put it on our diffusion cells. But most of the time, when we do our in vivo studies, we use the pig, because pig skin is very similar to human skin in that it's the same thickness. It has 4 to 5 epidermal cell layers, unlike mice or rats that have only 2 cell layers, and it has a nice thick stratum corneum. Also, the hair follicle density, and this is very important in nanoparticle studies, is that human and pig have 11 hair follicles per centimeter squared. And I want to point out to you that this is based on adult pig, and I'll tell you why, because young pig have more hair follicles per centimeter squared. So if you look at invagination of a hair follicle, is just this epidermis coming in. And if you did a cross-section of this here, you would most probably say that particles could penetrate in a hair follicle, but that's not true because it's the area of this hair follicle that has a breached epithelium. The cells are a lot more crumbular and more subjective to particle penetration. And this is a potential route of how nanoparticles may be getting in skin, is through these infundibulum areas of the follicle. So we studied different species and looked at normal rat. We tape stripped 10 times, which we got rid of that major rate limiting barrier, which is the stratum corneum. We measured reflection and abrasion, where we removed most of the epithelium. And we evaluated this with QDOT 655, which are the carboxyls. And you can see a normal skin, even though you have clipped the animal 24 hours prior, you still get the stubs of hair when you magnify it. So which particles don't sit on the skin, but they sit on the hair. We measured reflection. You get a little bit of more homogeneous dispersal, but with tape strip, you remove all that hair, and your particles gets to sit directly on the skin. And with abrasion, you must abrade it not till it bleeds, but just remove that upper epithelial layer. We also did this with pig skin. Again, even tape stripping 30 times, very difficult to remove that rate limiting area. And we also braided it 60 times with sandpaper, in which we could remove the stratum corneum layer. We also did this with human skin from surgical abdominoplasties and tape strip. You do get some corneum stuck in this area, and so it's very important when you're studying the penetration of nanomaterials, because it's not always homogeneous, and this is abrasion of removal of the epithelium. So when we looked at the three different coatings, the PEG, the pegamine, and the carboxyls, of these Q dots sitting on the skin, this is tape strip, we saw that even with removing that rate-limiting barrier, that again, your Q dot sat directly on the surface of the skin. Same with the pegamines. When the skin was dermatoned and placed on diffusion cells, they remained for 24 hours on the surface of the skin. And again, if there is an invagination of the hair follicle, this is really just a stratum corneum coming down. And if you did a cross-section, you would say it's in the hair follicle, which it really isn't. It's inside of the stratum corneum invagination. We also looked at it with rat skin, and we showed that this is what typical rat skin, it would sit on the surface over 8 and 24 hours. This with flexion tended to bind to the hair itself, and with abrasion, again, you see that without the epithelium, of course, particles can penetrate and get into the dermis. Then it would be capable of getting into the systemic circulation by getting absorbed in the capillaries and the dermal papillary layer. Again, here it is showing around the hair follicle. We also work a lot with drug delivery studies, and what we do at times is that you use penetration enhancers, and penetration enhancers can alter the lipid organization of the stratum corneum. We use some terpenes of methadol, eucalyptol, and limonene, and these are regarded as safe according to GRAS. But what we did is we took this human skin, we pre-treated with these terpenes to see if we could increase the penetration of these nanoparticles. Again, playing with different formulations. And what we saw here with just control and water, this is DIC, fluorescence, and fluorescent overlay, is that even with the penetration enhancers that tend to come in through the stratum corneum, and TEM just showed a little bit deeper in the stratum corneum layers, but we were quite surprised that we didn't see full penetration of these nanoparticles with penetration enhancers of terpenes. We also did studies with C60, which is much smaller, in which we took the C60 and put in these different solvents, such as an occupational scenario where you're working with different formulations of chloroform, cyclohexane, toluene, and mineral oil. And what we saw here was that when C60 was in chloroform, dosed every day on pigskin, then tape stripped and analyzed by simple liquid-liquid extraction, you could see that the first 5 or 6 tape strips, that chloroform penetrated, the C60 penetrated in the chloroform mixture much deeper than these other solvents. Okay, we also studied Q dots 565 and 655, which is, you know, when you purchase them, they say it's about a 4.6 cadmium selenide core with a zinc sulfide shell. When you put hydrophilic coatings on it, you then increase its size. We studied this spherical shape in this elliptical shape in pig skin, and what we saw that it rapidly came through both the PEG, the pegamine, and carboxyls. So size here was not an issue. It's the coating. And also if you look in the pegamine and amine of the 655 carboxyls, it took 24 hours before the carboxyls, which was the smaller diameter, hydrodynamic radius, and that took longer to penetrate. So again, surface coating played a major role in penetration. But I want to tell you that we did these in weanling pigs, and weanling pigs here have 68 hair follicles per centimeter squared, compared to the adult pigs that Bob Bronoff had reported on, about 10 hair follicles per centimeter squared. So again, we think that hair follicles may be playing a role in explaining how these Q dots are coming through in the weanling pig skin and not in the human skin as we had shown. So today we know that skin cancer is 1 million cases are diagnosed annually that are non-melanomas in the US. And now we know that some of the nanomaterials, such as titanium and zinc, are included in cosmetics and also in sunscreens. And older sunscreens give you this white, patchy appearance, but the nanoparticles in sunscreens will give you a clear appearance. So we wanted to know that if you had UVB sunburned skin, you topically apply more sunscreen with nanoparticles, would you see an increase in penetration? So we did UVB studies on pig skin, and we calibrated this. We first find it's minimal erythemic dose. We've done this for 10 years on UVB studies with cosmetics, and after 24 hours, we then removed the skin and dosed with the zinc and titanium formulations. These formulations consist of 10% rutile crystalline TiO2 in oil and water formulation. We also did it in a water and oil formulation at 10 by 50 nanometers. We also evaluated it in the zinc and coated with a triethyloxycaprylsilane and also uncoated zinc in different formulations. We then did TEM to look at the size of these TiO2 particles. We also looked at the zinc particles to show them in formulation. And we then dosed on the skin. We did DRACE testing, and what we saw with UVB exposed skin, you got a typical +3 because 48 hours you get after the sunburn, you get more of exaggerated response. And all of these formulations that was given to the pig did not increase that erythemic response anymore. We also did in vivo studies on the skin where we looked at the titanium with no treatment versus UVB, but UVB had caused penetration of the titanium particles deeper down these stratum corneum layers. They can be anywhere from 30 to 60 stratum corneum layers in the skin, and that can vary on different body sites. We also looked at TiO2 in this formulation and saw with the water and oil and water, that there is tended to be a difference in further penetration. We also looked at with zinc. Zinc did not penetrate as much as titanium by TEM. And again, here it is, zinc on the surface normally, with UVB still sits on the surface. We also looked at the agglomeration of the substance around the hair. Again, remember the hair, if the area is breached, that's a particular area where the cells are crumbly, the barrier is then broken, and that could increase the penetration of particle entry. Again, we looked and did EDX analysis to show we did have TiO2. Titanium, we did the same for zinc, and again, we show we did have zinc in our formulation, and that was bound to the hair and in this base of the hair follicle. So now we went on to do some more sensitive techniques, such as time of flight secondary ion mass spectroscopy, where here you have a business ion that erodes the surface of the skin, causes the particles and atomic mass to go up this time of flight, and then measured in a detector. So what you have here is a complete mass spectrum is acquired and stored at each pixel for what particle you're studying. And we did this for titanium and zinc. So what we show here with normal UVB skin and just normal skin treatment with these formulations, you could see that in vivo, that the titanium, we could see further penetration, more so that was shown that by TEM. And also here you could see with UVB, it sat pretty much into the epidermis. So there wasn't that much difference. If you look over here, you can see we had some hot spots, and these were not homogeneous, and I think these could be hair follicles because they're in the right location and about the right size, again, where particles could come through even in normal healthy skin versus UVB-treated skin. We did the same with zinc. Zinc stayed pretty much on the surface. Again, still pretty much on the surface. We also evaluated this in vitro because it's not always feasible to do in vivo, so could we come up with the same results in vitro? And again, we did the same type of studies, and we took the UVB sunburned skin, dermatome, put it on our diffusion cells, and did these specific techniques on them. And what we saw measured by ICP-MS, that the titanium was below the level of detection. That is when we put this on the surface and the flow-through cells and try to measure the uptake of titanium, which is a heavy metal. And what we saw was below detection limit in both formulations. And if you looked at the zinc, we did see some concentrations in 0.5 microgram per mil, but that was not suspected because we know that zinc is ubiquitous in the lab. It's also found in your powdered gloves, your glass, and your stainless steel. So, but it was still below background level. We also showed that the skin did get sunburn. We have typical pygnotic sunburned cells. And when we evaluated this in vitro, you could see TiO2 that sits on the surface here, and it still remained on the surface here with UVB treated. We did the same here with this formulation and see it on the surface. It came down in normal skin, but with UVB treated, it came down about the same. So in vitro tended to show a little bit more penetration. We also did that with zinc, which zinc was very difficult to localize, and we just saw it on the surface. Both formulations remained on the surface. When we did this with the TOF-SIMS analysis, we saw that with, this is normal UVB. This area here is treatment with normal skin, showing the penetration of TiO2, again, with all hot spots being hair follicles, and again, we saw some here. But TM had showed it a little bit different. Again, here with the next formulation, we did oil and water, water and oil. We could see hot spots here. This represents about 100 microns, so this is just below the epidermis. And again, the same here, which we saw penetration of titanium. Now, zinc was a whole different story. Again, we saw zinc sitting pretty much on the surface. Zinc did not penetrate very well, and here we did see it penetrate a little bit into the epidermis. So if you compare both the in vitro and in vivo studies with TiO2 and zinc, we saw titanium was more persistent than zinc. We also showed that by EDS, titanium penetrated into the stratum corneum in the in vivo and in vitro studies. Zinc penetration tended to be greater in the stratum corneum in vivo than in vitro. TOF-SIMS data of titanium zinc showed some penetration a bit in the epidermis. There was slight evidence of TOF and TIMS at 48 hours, and in vitro that penetrated through the dermis into small focal hot areas, and also perfusate samples, which is where you measure how much can come through of the heavy metal of titanium or zinc into the perfusate, was below and did not detected by ICP-MS. So we could say that there was really no absorption or flux of titanium or zinc in the perfusate. So important considerations when you assess nanomaterial absorption and penetration is that we showed that there are vehicle effects, and that if you put it in different formulations of surfactants, especially with the solvents like the chloroform C60, you could see that you had increased penetration. So these solvents can modify the particle structure, solubility, and agglomeration, as well as the interactions with the skin. We saw species differences in both rat, pig, and human, and the skin preparations and the age of the animal could also have an effect as to the rate of penetration of some of these particles. So, and also we have to be careful in that it's dangerous to try to extrapolate nanomaterials, as we did see some penetration with the very small FITC label fullerenes. The C60 at 3.5 nanometers did penetrate. Baroli in Italy has shown the same with ferric oxide, published in the Journal of Investigative Dermatology, where she saw small penetration, small molecules penetrating. So all studies are a function of particle size distribution, shape, surface coatings, pH, and vehicle, and penetration. Remember, it penetrates to skin but not necessarily gets absorbed, is a function of nanoparticle type, species, and skin treatment, whether it's been used as flexion, as we showed, which increased the penetration, mechanical action, such as we did with abrasion, or with UVB treatment. So I'd like to thank my lab and graduate students, EPA, 2-star grants in support of this research, also the U.S. Air Force for the aluminum and silver nanoparticle studies, the National Academy Keck Futures Initiative grant, and the NIH R01 grant on comparative nanomaterial toxicity. Thank you.